Okay, so we have completed mitosis successfully and we will switch on to meiosis. Meiosis is a very interesting division. Okay, it is not as easy as mitosis, but it is very interesting. So if you understand meiosis, then it's like you might feel it is little bit confusing also, but if you understand with basic, meiosis is also very easy. Okay, let us start the next division, and that is meiosis division. Okay, first of all, what we'll understand in this is that some key points. Okay, what is meiosis? We have already discussed. But let us understand some key points and then we will understand those key points in detail. The key points are like this. The first thing about meiosis, it is reduction division. Yes or no? Do you agree with me? What it means? It means if a parent cell is having any number of chromosomes like 16 for example, if it is diploid, then it will produce how many cells? It will produce 2 cells first of 8 and 8. Then it will produce again 2 cells. Here again 2 cells. Totally how many? 4 cells. What will be the division and what will be the number of chromosomes? It is the same as this. So this is complete meiosis. This part is meiosis 1. This part is meiosis 2. Okay, every time you should note this, that meiosis is a reduction division. It completes like this only. This is how it works. Whatever the number of chromosome you have in the parent cell, you just make it half and it will produce 4 cells. We know that also. So it is reduction division. I hope it is clear. Second key point, where it will occur. Where it will occur. The cells which undergo meiosis are called meiocytes. M-E-I-O. Meiocytes. What are meiocytes? The cells. Now this cell, if it is undergoing meiosis, then this cell is called, it's called meiocyte. Check out with the spellings, okay? It's called meiocytes. So what are meiocytes? Meiocytes are those cells which undergo meiosis. Okay, this is clear. Now, the next key point about meiosis. Where it occurs? It occurs in meiocytes. Now what it produces later after meiosis, what is the product? What it produces? It is important for the formation of what? Gametes. Why meiosis is important? Because it helps, it undergoes, the meiocytes will undergo meiosis, and what they produces at last? They produces the 4 gametes. These are the product. So the product of meiosis is gametes. How many? 4 gametes. Okay. Now, finally, again, what you need to understand here in meiosis is that, okay, so it helps in the formation of gametes. It is reduction division. It takes place in the meiocytes. I hope this much is clear. Then, totally it produces what? From one parent cell, it produces 4 gametes. This is how it works. It's not sure that all the 4 gametes are functional. That is different thing. But meiosis works like this. Okay? Okay. Next, what we will see about meiosis is that little bit in detail about those. Okay? One more thing here. See here. Meiosis always, like just now we have discussed about mitosis. That mitosis, there are exceptions wherein mitosis will take place for haploid cells as well. But meiosis is not that condition. Whenever meiosis is happening, obviously the cell should be diploid. That means meiosis happens only for diploid cells. Why? Because it is producing haploid cells. Then obviously it should work on diploid only, no? So meiosis occurs always for diploids. Only diploid cells can undergo meiosis to produce what? To produce haploid cells. Diploid cells will undergo meiosis. Why? To produce haploid cells. And those haploid cells we call them as gametes. We call them as gametes. I hope this much is clear. This is very crude thing about meiosis. Now, why this meiosis is very special compared to mitosis? That is what we need to understand. Speciality is because of reduction. Chromosome number is reduced. Okay, what is the importance of meiosis? That we will discuss at the end of this concept. But now what we need to understand is how it is important, like when we comparative analysis, mitosis and meiosis. Comparison, I think this all things are comparison only. One more important comparison, what we need to, before that, take down this much. Okay, now, okay, I'm continuing this. Okay, how many? 1, 2, 3, 4, 5. These are 5 points I have written here. Still we need to continue. Okay, what are the next points? We will see. Comparative analysis we are doing of mitosis and meiosis. The next key point for meiosis is that meiosis takes place, I mean, it's not in a one go. It will take place in 2 phases. So it works like meiosis 1 and 2. There are 2 meiosis, meiosis 1 and meiosis 2. When this meiosis 1 and meiosis 2, both of them completes, then meiotic division is completed. Mitosis, in case of mitosis, that is not the case. Mitosis one step, in one step only, you will find phases like prophase, metaphase, anaphase, telophase. Those phases you will find mean meiosis also. But meiosis have divided into 2 phases, meiosis 1 and meiosis 2. So from here till here is meiosis 1, from here till here is meiosis 2. Plus 1 plus 2, totally we call it as meiosis. Okay, so this is one thing. Now one more thing, this is very very important. Comparative analysis of mitosis and meiosis, if you consider, there are 2 important things which happen here in meiosis, that is homologous chromosome pair up. And another important key point of meiosis is crossing over and recombination of genes. This is very very important. These are very important points which we need to discuss in detail. Okay, these 2 I am going to discuss now in detail. Before that, the last point, the key point for meiosis, one more, is that, okay, so here about the, what you can say, no, okay, before telling you the statement, I would like to ask you one question. When we were discussing cell cycle, we know that cell cycle is divided into interphase and M phase, and there we know that M phase is not meiosis. Why it is not meiosis? Why it is only mitosis? Because mitosis is a division wherein parent cell divides and forms daughter cells. Those daughter cells also can divide and undergo the same process. So in cell cycle we have mentioned M phase is mitosis phase only. Then what about meiosis? Is meiosis a cycle? Meiosis division is not a cyclic division. It is not, it doesn't works in the form of cycle, because once the parent cell divides and forms the gametes, these gametes are used to fuse with other gametes, opposite gametes. It will not divide again. It will not go to the same cycle again. It will not run the same cycle again. So it is not involved in cell cycle. But what about the interphase in meiosis? Does meiosis undergo interphase or no? Directly it will divide. The cells should not, the cell which is dividing, which is undergoing meiotic division, is nothing but a diploid cell, no? How it has developed? It has been developed by mitosis, and then it has 16 chromosomes. Obviously it has to undergo interphase, wherein it again undergoes what? So in meiosis also you find interphase. Okay, in interphase of meiosis also you have the 3 phases. You know which are the phases? G1, S and G2. Okay, and now you might get confused with this interphase and the meiosis 1 and 2. Where is this interphase working? Before meiosis 1, after meiosis 1, or after meiosis 2? How? That also is confusion now for you. So this two, just take down this much. We will discuss this and this now. Okay. So see here, we will discuss this two. Before that, we will discuss this one and finish it off. Okay. Okay. Interphase G1 and G2, meiosis 1 and 2. See here, any cell, which is undergoing division, it needs to prepare for the division. So for meiosis also interphases must. But that cycle, it's not in the form of a cycle. You can consider this is like this, this type of thing. So it will undergo interphase like this. Okay. Now let us consider this is interphase. Interphase is divided into what? G1, yes, and G2. So it is G1, yes, and G2 phase. And after that, when it starts meiosis, this is meiosis 1 and this is meiosis 2. See, in meiosis 1, what will happen? One parent cell will divide and form two cells, correct? If it is, let us take human example. If it is 46, which is a diploid number, it will form 23 and 23, which is haploid. In meiosis 1, what happens in meiosis 2? In meiosis 2, this will again undergo divisions to form how many? 4 cells. What is the number of chromosome? 23, 23, 23 and 23. It can be sperms, it can be eggs. Depends upon which diploid it is. Female or if it is male. So this completes meiosis 2. Once meiosis 2 is completed, it will not come back. These cells will not come back to G1, yes and G2. They are gone for fusion, for fertilization. So meiosis is not a cycle. It is this type of pathway, you can say, how it works. So this is interphase in meiosis again. So interphase works for meiosis, yes. Because meiotic cell, the meiocyte which is undergoing meiosis is a product from mitosis. Zygote divided and formed these cells. Now these also should grow. These also should replicate. Yes or no? Those things are important, no? Even the cytoplasmic content should increase. Because this cytoplasm should be given to all the 4 gametes as well. Nucleoplasm also should be divided properly here to form the gametes. It is not sure that it is correctly divided or no, like mitosis, but it should divide, no? So for all those things, G1 phase is important. For the replication of DNA, yes phase is important. And G2 is not that complicated like mitosis here. G2 is very small phase in meiosis. That is, okay, you can understand that interphase works for meiosis as well. G1 also works, yes also works, G2 also, very little bit, but this is how it is represented as. I hope I have cleared that interphase works for meiosis. Now the question is where, when. So here only you can understand, no? Before undergoing the division, before meiosis 1 only, interphase will be working. Okay, that means, for example, if you consider this cell, this is a meiocyte, which is diploid. It will undergo, it has been formed. How it is formed? It is formed by whom? It is formed by zygote only, no? That means it is formed by the process of mitosis. You know that very well. And this cell now, it will undergo G1 phase, S phase, G2 phase, and then meiosis 1 and then meiosis 2. So what you understand here is that, so before the start of meiosis, once only the interphase will work. It will not work twice. Some students think that after meiosis 1, again it will undergo G1 yes and G2 phase for meiosis to know. Please draw this diagram properly and be sure that interphase works before meiosis. Once the cell grows, DNA replicates, then it is ready for meiosis. Again, it will not stop anywhere. Continuous process it is. So before meiosis, one only interphase will work. If the cell undergoes interphase before it starts meiosis. Once it starts the meiosis, there is no any other phase which is interrupting meiosis 1 and 2. Interphase works before meiosis 1. I hope I made it clear. Now still, okay, take down this much. We'll discuss a little bit. Interphase starts from G1 till G2. After that it is meiosis. So we are talking about meiosis. Hope you people understood. Okay, to make this still more clear, okay, still some students will have some kind of confusions, all those things. I would like to make it completely clear. So we will go in detail little bit to understand this interface, why it works, how it works, whatever. Okay, see, with human example only I will give you, using human example. See, in humans, what will happen? Zygote will undergo mitosis and form many cells. And those cells which develop because of mitosis, it can differentiate and form so many cells. So let us take an example of spermatocyte. What is spermatocyte? Spermatocyte is a meiocyte. It is a meiocyte. What is its function? It will undergo meiosis. So zygote underwent mitosis to form spermatocyte. Now the spermatocyte will undergo which division? Meiosis. Why? To produce the gametes. Which are the gametes? Sperms. We call them sperms. How many? 4 functional sperms. If it is egg, 1 is functional, 3 becomes polar body because of unequal division and all those things. That we are not discussing here because we are concerned about some things which is confusion. So here what I am trying to say is that, see here, zygote underwent mitosis to form spermatocyte, and spermatocytes will undergo meiosis to form sperms. Clear? This at least is clear for us. Now what we need to discuss here is that, here I told you meiocyte, this meiocyte will undergo interphase. It will undergo interphase. And once interphase is cleared, like G1, yes, and G2 is clear, then it will go for meiosis directly. Okay, and the confusion for some students is why it needs to go this cycle. Why it needs to go to this interphase before division. There are some students who have this confusion. So let us talk about that. Okay. Okay. With the help of, I'll just show you. For example, you know mitosis. I'll show with the help of 4 chromosomes. Okay. Now this is a cell which will undergo mitosis. Let us consider this is the zygote. And in humans we have 46. Okay. Okay, fine. 46 only we'll consider. Okay, this is zygote. It underwent divisions to form what? To form spermatocyte. Obviously, spermatocyte will have 46 because zygote underwent which division? Mitosis. Now, what are these? Let's consider this is spermatocyte. Now, this spermatocyte, I told you, first it will enter interphase. right? And then it will, when it enters interphase, it will complete G1, yes, and G2 phase. Then it will come for meiosis division. Correct, Alva? Now the question is, why? Why this spermatocyte is going through interphase before meiosis only? That we are talking about. See here. Why this happens? Because, remember, the zygote, which has divided into 46 and 46, the chromosome, if you see how it looks, this is how the chromosomes are. Like this. I am not showing all the 46 here, okay? But try to understand, they are in this form with single chromatid. They all are like this. The spermatocyte in the earlier, the myocyte, it is a myocyte. The spermatocyte has how many? 46 chromosome in this form with single chromatids. When it will undergo meiosis, okay, it needs to divide and form sperms, no? That means in meiosis 1 and in meiosis 2 actually, the centromere will divide. So that time it should have this chromosome like this, no? It should have the another part. This is important. This is important for that. So, what spermatocyte will do? First only it will undergo G1 phase, wherein it will increase the number of cytoplasmic content. It will undergo S phase, wherein it will replicate the DNA in S phase, and in G2 phase is very small phase there, and then it will undergo meiotic 1 and then followed by meiotic 2. So meiosis is a continuous one. It will not take time in between. Meiosis 1 and meiosis 2 के बीच में there is no more time left over there. There it will not wait for replication of DNA. So before that only replication is done prior only. Okay. So that is why try to understand one thing. What we are discussing about here is that where interphase is there is before the start of meiosis. In mitosis also, before the start of mitosis, interphase used to work. Here also, before the start of meiosis. What students will have confusion is, they think that meiosis 1, first meiosis 1 will happen, then it will undergo interphase, and then it will go for meiosis 2. No, this is completely wrong conception. That's wrong. Interphase, meiosis 1, meiosis 2. Clear? Okay. Then, and one more thing I finalized that meiosis is not a cycle. That is why in cell cycle we did not talk about meiosis, and that M phase was mitosis only. Okay. Coming to the last one, the last things what we discussed about homologous chromosome, pairing, all those things, right? Okay. So, okay, I just want to explain that before going to the actual process of meiosis, because if you don't understand the concept of what is this homologous chromosome and what is recombination and all those things, you will not understand meiosis properly. So first we will try to understand what is homologous pair or what is homologous chromosomes, because meiosis, I told you, pairing of homologous chromosome. So to understand that, you should first know what is homologous chromosome, right? So we will discuss that. See here, what are homologous chromosomes? Like example of humans only, if you consider, in humans only, the sperm and the egg will fuse to form zygote. Correct, no? How many chromosomes sperms will have? 23, because it is a gamete. What about egg? Egg also will have 23. 23 plus 23 will be 46. So this 46 chromosomes what you have, this 46 chromosome, out of this, 23 is from sperm, 23 is from egg. Okay. Now this chromosomes which are given by the sperm, we call it as paternal paternal chromosomes. How many paternal chromosomes? 23. From mother, from egg, how many we are getting? 23. What we call them? We call them maternal chromosomes. Chromosomes from sperm are called paternal. Chromosomes from egg are called maternal chromosomes. So both paternal and maternal chromosomes combine together in the zygote. Okay. Now, so this 23 chromosome, if I start drawing all the 23, I cannot draw all the 23. I'll just try to explain you homologous. Those 23 chromosome, if you see, okay, this is how, let us consider this is the first chromosome. I'm just showing you 1 or 3 example, okay, with 1 or 3 chromosome. And let us consider this is the second chromosome, how it looks. And let us consider this is the, this is the third chromosome. See, you know that chromosome differs based on centromere position. Yes. So all this 23, and I cannot draw all the 23. I am just showing you the first 3. Same like this only, mother chromosomes, they are 23. No, there also you will have same type of chromosome. The first chromosome will be like this. It is similar to the first chromosome of fathers. Second is like this only here. Second one, they both are same. Third chromosome, they are almost similar only. I am just showing you, just trying to explain you. This is third. Likewise, now this first chromosome is having the same partner in maternal chromosomes also. Now the second chromosome is also having same partner in maternal. So if in paternal and maternal, whatever the 23 chromosomes they have, all the 23 have a pair. Both of them, like maternal chromosomes have a pair in paternal as well. Sorry, paternal chromosomes have a pair in maternal, or maternal have a pair in paternal. Means first chromosome is similar to this. What I mean by similar, it's not only about the diagram, what I have shown, the position of centromere, both are metacentric. It's, yeah, that is also one thing, how you can, they both are similar. They both are metacentric, so they both are similar. These are, if you see the little bit, it is away, submetacentric, so they both are similar. One more important thing is that, why they both are similar? Because they have genes, particular genes. Now for example, let us consider the first chromosome of paternal have gene in this particular locus. In this particular area, it has a gene, and let us consider this gene is for eye color. Eye color. Then same gene you will find here also. Means the color may be different. Now here it is, this gene is coding for which, what character? It is coding for eye color. It may be black. Eye color means eyeball color. It is black. Then the partner of this chromosome is this, no? Both are first chromosome, both are metacentric. Both will have genes at that similar place. Even this is coding for eye color. And let us consider this eye color is different. Let us consider this is blue. This is black and this is blue. What I am trying to say is that why these two are partners? Because position of centromere is also same in both. Main is that they have genes which code for similar character. It is having gene for eye color, the same gene even this will have. Which is strong among these both, or that is a different thing. Again, we will discuss this later. We call it as in particular locus. Locus means the area, the place where that gene is present. In the same place, this chromosome also will have same gene. That is why we call them as partners. So this chromosome have a homologous pair, and which is that homologous pair? This one. Now let us consider second chromosome. This chromosome is having somewhere here, okay, this has a gene which codes for height. Height, it may be tall, for example. Then the second, now this is second one, no? So same area in the second chromosome of the maternal from the egg, here also you will have the same thing for height. It may be anything. It may be tall. It may be dwarf. Let us consider dwarf. It depends. Clear what I am trying to say? So here what I am talking about is all the 23 chromosomes of paternal are similar with the 23 of maternal. But the last one, 23rd one, if I talk about, if it is a sperm, then obviously it will have which one? It will have Y chromosome. And you know, if it is XY chromosome, here if it is female, the eggs usually they will have what? They will have double X. And if it's 2X, father will have X and Y. The last pair is X and Y. Here it will be X and X. Likewise, what will happen is that in this zygote, if this zygote is having X and X, then it leads to what? Female. X and Y will lead to male. Again, we are not going for sex determination here. We are just trying to understand that all the chromosomes which are present in sperm, they have their partners in the egg also. So when these 23 and these 23 come together in the zygote, then they will have this pairing, homologous pair. We call them homologous pair. So this is a homologous pair of this. This is a homologous pair of this. The last one is third one is a homologous pair for the third one. I hope I made it clear what is homologous pair. So when this zygote is undergoing meiosis, in that we are talking about this homologous pair in that condition. So there we will talk in detail, but you just understand, I hope you understood what is homologous pairing. These are homologous chromosomes. Clear? Okay. Now there is one more thing we will talk about. First take on this match. Okay. So this is about homologous chromosomes. Okay. Now let us talk about one more concept which we will be discussing in meiosis. So we have done homologous chromosomes. Now we will discuss something called as crossing over. Crossing over. It is also called as recombination of genes. Recombination or crossing over. See, I'll just explain you here only, because later on when I am discussing about meiosis first and meiosis 2, there I'll be using these terms. Crossing over, all this, homologous, all this thing. So you should know that. Now let us consider, this is a chromosome. Okay. This is a chromosome from sperm. And the same chromosome you will find from mother, that is from egg. And let us consider this is a one. This also will have this type of structure only, right? So these are what? This is from sperm. This is from sperm. I am talking about zygote. So this is from sperm and this chromosome is from egg. Okay. Now this might be any number of chromosome. Okay, that doesn't matter. That doesn't matter. It might be here, it might be anywhere here, but these two are similar, no? So these two come together. And when they come together, we call them homologous pair. What do we call them? We call them homologous pair. And already when I taught you about the structure of chromosome, I have told you that even if you consider this diagram like this, this is a chromosome. This is a chromosome with single chromatid. This complete from here till here, what it is? It is a chromatid. Now this also is a chromosome, this also is a chromosome. This is having two chromatids which is identical. This and this are both same. This and this both are same. So if this will have another copy, this we call them, both of them are sister chromatids. Correct, Alva? These two are sister chromatids which have single centromere. If you consider this, then tell me the sister chromatids of these two. This A and B, or A, let us consider this complete chromatid is A and this complete chromatid is A1. These both are sister chromatids. And here if you consider this one, this and this are sister chromatids like B and B dash. These both are sister chromatids. So I will write it here. Sister chromatids. Which are sister chromatids here? The first one, like A and A dash, and in the second one we have here B and B dash. These both are sister chromatids and these both are sister chromatids. So you know what are sister chromatids. Earlier only I have explained. Now let us consider when these two homologous pair come together. Tell me this A dash and B. What are these both? What are these both? Do you think they are sister chromatids? No. They are non-sister chromatids. They are non-sister chromatids. So please try to understand. Whenever there is a pair, whenever there is a homologous pair coming together, that time one chromatid of one chromosome of the homologous pair and the other chromatid of other homologous pair. I mean this and this. So this chromatid and this chromatid, these both are not sister chromatids. We call them non-sister chromatids. So if I write example, you'll remember. Example, this is A dash and this is B. So A dash and B. A dash, I've written with black, so I'll be writing with black only. A dash and B are non-sister chromatids. What about this A and B dash? Even these are non-sister chromatids. A and B dash are also non-sister chromatids. So A I have written with black. So A and what? This B dash. So this is a homologous pair. Yes or no? These both together, we call them homologous pair. Because this pair is, this chromosome is from sperm. The same type of chromosome we have in egg also. So these two they can form a pair and we call them homologous pair. But in a single chromosome, we have two chromatids and those two chromatids are called sister chromatids. In this also we have two chromatids. These two are also called sister chromatids. But the opposite chromatids of these two pair, I mean this one pair here, this one and this one, if you see, they are non-sister. And this also, they are non-sister. I hope you understood what are sister and what are non-sister. So what about these two then? What about this and this? Even these two are what? These two are non-sister chromatids. Okay, I hope this term also is clear. Now we are talking about crossing over, no? So I will tell you what is crossing over now. So what will happen in this crossing over means non-sister chromatids will overlap with each other. I will show it here. So this is one chromosome like this. This is a non-sister chromatid. For what? If I consider this chromosome like this, so this and this is a non-sister chromatid. Now what is crossing over means? What will happen is that this non-sister chromatids, okay, I'll show you what will happen. These non-sister chromatids will, they cross like this. When they come together, what they do is they cross like this. I'm just showing you. Okay, example. What happened now? You can see that non-sister chromatids, this and this, crossed. They overlapped on each other. So this is called crossing over. So what is crossing over? Just you can see like this. So these two are non-sister chromatids. This is from one hand, this is from one hand. These both are crossing. So cross between whom? Cross between non-sister chromatids. Now if these two are chromatids of the same chromosome And if these two will pair, then it is pairing between sister chromatids, which will actually not happen. Always pairing happens with non-sister chromatids. So this is called crossing over. They crossed with each other. Okay, then why do we call it as recombination? Because when these two chromatids or whatever, it can be like this also, huh? Like for example, complete crossing over means you can consider like this also. It is like confusion, so I have shown it like this, and that means complete crossover between both the non-sister chromatids. These both also, like this. They can overlap on each other and they can cross. So this is called crossing over. Why do we call it as recombination? Because whenever they cross, for example, now let us consider. So this have been crossed over this, no? Try to understand. This is completely the blue colored is a anything. It can be maternal, it can be paternal, that doesn't matter here. What we need to understand is this complete chromosome is of blue. I'm just explaining you why we call it as recombination. So this chromosome is black, like this. With the help of color, I am trying to explain. That's it. Like this. Okay. So what this is called, what this process is called? Crossing over, overlapping of non-sister chromatids is called crossing over. But once the crossing over is done, what will happen is that the genes which are present in this part, which part have been crossed, the genes which are present in this one is exchanged with the other one. That means when you separate these two chromosomes, how you will able to see the chromosome? I'm showing what will happen later on. You will see that chromosomes after separation, you will be able to see that these will have particularly the genes of this chromosome over here, because there only they have crossed, and this here. Same thing you will find it here also. This genes have been copied by this, copied in the sense completely gone for this one, like this. Are you understanding this one? Yes. So once the once the crossing over is done, then you will find this type of thing, wherein this chromosome is not completely paternal now. This chromosome is not completely maternal now. Why they are not completely paternal and maternal? Why you cannot call them now as paternal and maternal? Why? Try to give the answer. Now you cannot call them as paternal and maternal because they are not similar now. This paternal is having some amount of maternal chromosome. These two segments, can you see? And this maternal is having some amount of paternal. These two. Why this happened? This happened because of crossing over. And what we call this as? We call this as recombination. So what happened was the genes of this chromosome and this chromosome have exchanged. Because of that, what happened? There is a process called recombination of genes. Recombination. Are you understanding this term? Recombination. From this part, the genes went and combined to this chromosome, and the genes from this chromosome came and combined with this one. Why? Because of crossing over. So this is called recombination of genes. This and the pairing. Why this happened actually? This happened because these two chromosomes came together as a homologous pair. So homologous pairing and crossing over, these two are the very important part of meiosis. That is why meiosis is so different than mitosis. All the sperms what are produced from meiosis, they all will not have same ditto, like exact copy of father or mother. That is why the children of parent, we are not exact Xerox of our parents. We are mixed. We have characters of father, we have characters of mother as well. Why? Because this is the reason. And this is called, variety, variation. This is how variation is produced. And always in sexual reproduction only you will find variation, because in sexual reproduction gametes are produced, and gametes are produced by meiosis. And meiosis have this two important concept of crossing over and recombination and homologous pair. That is why it will give you variation, and that is why meiosis is important. I hope I made clear what is homologous pair, what are sister chromatids, what are non-sister chromatids, pair happens between whom, and what is crossing over and what is recombination. So it is a very basic thing. Now we will start meiosis next. So please remember all these things. Thank you.